Hey folksies, in this video we'll talk about HPLC. So let's get started. HPLC is known as high performance liquid chromatography. We also can call it as high pressure liquid chromatography. Its cost for analysis is too high, that's why we also can call it as high priced liquid chromatography. Now if you see the definition, then HPLC is a chromatographic technique that is used to separate a mixture of compound in analytical chemistry as well as in biochemistry. It generally does its job for identification, quantification and purification of individual component of the mixture. This photo represents the basic arrangement of a HPLC. Commonly used principle in HPLC is separation based on polarity. In this technique, separation occurs depending upon the distribution of analyte between mobile phase and stationary phase, where the polarity plays a vital role. That is, if the mobile phase and analyte both are polar, then they will attract each other and they will elute later. Whereas if they are of opposite polarity, then they will repel each other and will elute faster because of their weak attraction force between the molecule. Physicochemical characters of a molecule is generally determined by its atoms and the bonds between the atoms. So these two factors generally determines whether a molecule is polar or non-polar. Unlike a magnet where the opposite pole attracts each other, a molecule with similar polarity generally tends to attract each other, and those with dissimilar polarity may sometimes repel each other. So we can remember this mechanism by making a slogan of like attracts like, that is, polar will attract each other. Now, to design a chromatographic separation system, we generally have to make a competition between the various compounds that are present in the sample by choosing a mobile phase and a stationary phase with different polarities. After that, the compounds in the sample that are of similar polarity to the stationary phase will be delayed because they are more strongly attracted to the particles, whereas the compounds whose polarity is similar to the mobile phase will be preferentially attracted to it and move faster. So if you see the heliotropic series, then it is nothing but a scale upon which the common solvents are placed in order to their relative polarity. In this picture, you can see the inventor of HPLC, who was a botanist by profession and wanted to study the leaf pigments, mainly chlorophyll. That's why he invented HPLC. Now let's see the various types of HPLC separation. First one is normal phase, where the stationary phase is polar and the mobile phase is non-polar. If we see the figure, then it represents a normal phase chromatographic separation where we use 3 dye, from which the yellow dye retains strongly because it matches with the stationary phase polarity, and the other 2 dye, that is red and blue, matches with the polarity of mobile phase but does not matches with the polarity of stationary phase. That's why they will elute faster. Our next chromatography separation is reverse phase chromatography, where the term reverse phase generally used to describe the mode of separation that is completely opposite to the normal phase chromatography, where the mobile phase used is polar and whereas the stationary phase is non-polar. Figure shown above describes the separation based on reverse phase chromatography, where we can see the blue color dye retained more strongly because it's non-polar and the stationary phase is also non-polar. On the other hand, the polar yellow dye, being weakly retained, is won in the competition because it's polar and the mobile phase is also polar, so they eluted earliest. Our next technique is ion exchange chromatography. In ion exchange chromatography, the mode of separation is based on electrical charges. The rule here is reversed. Here, the like charges may repel each other, where the opposite charges attract each other. So the cation exchange is used to retain and separate positively charged ion on a negative surface, whereas anion exchange is used to retain and separate negatively charged ions on positive surface. Our next chromatographic technique is size exclusion chromatography. In 1950s, Pratt and Flodin discovered the biomolecules could be separated based on their size. by passing or filtering them through a controlled porosity hydrophilic dextran polymer. This process was termed as gel filtration. Later on, organic polymer packings were used in place of hydrophilic dextran polymer, and that is used for separation. Similar separation done using controlled porosity silica packings were called size exclusion chromatography. Now let's see the instrumentation of HPLC. High pressure liquid chromatography. As the name only suggests that it needs a high pressure, and for the generation of the pressure we need a pump, and this pump is also able to generate a consistent pressure and a controllable and reproducible flow rate. Next is injector. An injector is generally used to introduce the sample in the flow of eluent. The simplest and easiest way to introduce the sample is by using a syringe. The most widely used injection method is based on sampling loops, but nowadays generally autosampler is used, which allows us to repeated injection in a set of scheduled timing. The separation of the sample is performed inside the column. The recent columns are often prepared in stainless steel housing instead of glass column. The packing material that is used inside the column is generally silica or polymer gel, and the eluent used for liquid chromatography varies from acidic to basic solvents. Most column housing is made of stainless steel because it can withstand high pressure as well as it can tolerate wide variety of solvents. A detector is generally used to observe the obtained separation inside a column. The composition of the eluent is consistent when no analyte is present in it, but the presence of an analyte changes the composition of the eluent, which detector measures and gives electrical signal. The electrical signals detected by a detector is not visible to our eyes. That's why we generally use a specifically designed software which has the features like data acquisition, peak fitting, baseline correction, automatic concentration calculation, molecular weight determination, etc. Invisible gases such as oxygen can be present in the eluent and can cause a noisy and unstable baseline. That's why degasser is used, which are special polymer membrane tubing to remove gases from the eluent. Column heaters are generally used to keep consistent temperature conditions of the column. In liquid chromatographic separation is often largely influenced by the column temperature. Sometimes for analyzing such as sugars or organic acids, better resolution can be obtained at elevated temperatures, that is 50 to 80 degrees Celsius. Thanks for watching this video, and if you like the content then do subscribe my channel and hit the like button.